Hello, my name's Antonio.
And my name is Amalo.
Hi, I'm Briseida.
Hi, I'm Giselle, and for this project, we are going to talk about horizontal gene transfer. So we have different resources. We have two articles, Article 1 and 13, that are being pulled from this class. And we also have two more outside resources that we will tie in all of the articles together. We will be going over these articles, especially the two outside resources, where we will show different figures and explaining it in depth while trying to tie them all together. So in this first article, the evolutionary consequences of drug resistance, shared principles across diverse targets and organisms, talks about how bacteria was able to gain resistance for the antibiotic. It states that there are different ways to obtain resistance: natural mutation, viral genetic information, and horizontal gene transfer. In this article, it talks about horizontal gene transfer and what it can do. Horizontal gene transfer is the movement of genetic information between two organisms. In the article, it also mentions that in order for gene transfer to occur, it does require close contact. Since horizontal gene transfer involves exchanging genes, it does mean that the bacteria can gain antibiotic resistance and a higher fitness cost. Stated in the article, there are 4 different factors for horizontal gene transfer to occur. The first is the presence of resistant genes in the total rhizostome. Second, the ecological opportunity for horizontal gene transfer. Third, the efficiency of transfer versus genetic barriers. Fourth, the fitness effects of resistant mechanisms on the recipient. This is a powerful and important mechanism that will be further explained in the next article, Article 13.
Article 13, lateral gene transfer and the nature of bacterial innovations, is about lateral gene transfer, which is another way of saying horizontal gene transfer, and how it plays an essential role in both the diversification and similarities that you find between bacterial organisms. The article mainly focuses on methods for detecting horizontal gene transfer through ways that are other than visual. As such, it would be them searching through the genome to find similarities and differences between bacterial organisms they presume may have undergone horizontal gene transfer. So, horizontal gene transfer is a mechanism for both diversification and convergence. What I mean by that is that it diversifies bacterial species because horizontal gene transfer can happen between different species of bacteria. We're able to see different plasmids being transferred, different genes and different traits being transferred between species of bacteria that would otherwise not be closely related. And because of that, the genome sequence becomes changed and you get a more diverse gene pool. What I mean by it is a mechanism of convergence is that because that gene pool is picking from the same set of plasmids, you'll find similar genes and traits among the bacteria, keeping them closely related in some form. So within horizontal gene transfer, there are 3 different paths that it can occur in. As you can see in the diagram, there's transformation, conjugation, and transduction. Each pathway is dictated by the method in which the donor DNA is acquired, and that is precisely how it's classified. The first one we're going to talk about is transformation. Transformation is the uptake of naked DNA by a prokaryote through the means of simply taking the DNA through a transmembrane protein. The donor DNA in this case is free-floating DNA from spliced cells. As you can see from the diagram, we see that naked DNA from a lysed donor cell is absorbed through transmembrane channel proteins of a competent cell, the recipient cell, and the naked DNA is then combined with the chromosomal DNA, and those genes associated with that DNA are able to be represented in the bacteria. So the second method for horizontal gene transfer is conjugation. Conjugation is a process by which 2 prokaryotic cells manually conjoin together via a structure known as a pilus. As you can see from the diagram, it is quite literally 2 bacterial organisms connected by a pillar, and they're able to transfer plasmids between each other. And this is very much similar to a sexual process, but it's different in the way that there's no offspring produced, but rather the changes are implemented then and there if the plasmid is absorbed and integrated into the bacterial's chromosome. So the last way that horizontal gene transfer can occur is through transduction. Transduction is the process by which a viral bacteriophage attaches itself to a host cell and injects the viral DNA into the bacterial host. And the bacteriophage DNA will then be integrated into the bacterial chromosome. And from there, it will utilize its reproductive functions to create more of the bacteriophage and cause the cell to lyse and die but it will spread more of the bacterial DNA, causing an endless loop, as you can see from the diagram. Okay, so now that we know what HCT is capable of and how it works, it is essential to go more in depth on how the mechanics cause problems and benefit society in our modern day world.
Okay, so one of the outside sources that we chose is titled Horizontal Gene Transfer and the Genomics of Enterococcal Antibiotic Resistance. The researchers focus on how bacteria are able to transfer antibiotic resistance to other bacteria through horizontal gene transfer. So this paper focuses heavily on the bacteria Enterococcus, and the reason for this is because it is known to cause antibiotic resistance in hospital infections. And through horizontal gene transfer, it is able to pass its antibiotic resistance. This process is mediated by pheromone-responsive plasmids. Enterococcus is a very interesting bacteria because it is found in different organisms. For example, it's found in the gastrointestinal tracts of prey and predators. Therefore, the bacteria needs to be able to adapt to their different environments. So because prey and predators have different diets, the Enterococcus bacteria need to be able to adapt to the different nutrients. And so pheromone responsive plasmids are known to be found in the species of Enterococcus faecalis. As the prey and predators E. faecalis strains commingle, they are easily able to adapt to any particular host and their dietary habits. And every plasmid contains specific receptors that sense different pheromones. For example, the E. faecalis has a plasmid PAM273 that senses a specific signal or pheromone. In this figure, we can see how E. faecalis is able to donate the antibiotic resistance through the help of pheromone-responsive plasmids to a different bacteria. In this case, it is the S. gordoni chalis receiving antibiotic resistance. In section A, we can see how the S. gordoni chalis bacteria releases the Gordoni CAM373 pheromone that is detected by the E. faecalis that is carrying the plasmid PAM373. In section B, we can see how antibiotic resistance plasmid is transferred to S. gordoni chalis. We can see how the pheromone induces a plasmid, which induces the transfer of the antibiotic resistance plasmid. This process leads to S. gordoni chalis to obtain antibiotic resistance towards erythromycin. It is also important to notice that if the E. faecalis bacteria didn't contain the pheromone responsive plasmid PAM373, then it wouldn't be able to transfer the antibiotic resistance to the other bacteria. And in this figure, we can see how antibiotic resistance is transferred in different ways. So here the red line represents the vancomycin resistance gene. In section A, we can see how plasmid PAM830 induces a transfer of the resistance gene to S. aureus. There are two possible outcomes. Either the plasmid PAM830 is transferred to the resistance gene, transferred into the chromosome, or is transferred into the chromosome of the receiving bacteria. In section B, we can see how the resistance gene that is located in plasmid PAM830 was not able to fully transfer to the recipient bacteria. In this case, they need a native plasmid, which is PSK41, to transfer the gene. And so now that we have an idea of how antibiotic resistance is transferred through horizontal gene transfer, it is also important to understand how we can use other methods for gene transfer using CRISPR.
The article CRISPR-Cas immune system and genetic transfer reaching equilibrium demonstrates how bacteria have their own mechanisms such as clustered, regularly interspaced, short palindromic repeat known as CRISPR, which correlates to CRISPR-Cas genes. The CRISPR-Cas system regulates transcriptional factors, pathogenesis, DNA repair, sporulation, and the modulation that produces biofilms. The constantly updated mobile genetic elements in transposons and plasmids are usually associated with the transmission of antibiotic-resistant genes. The CRISPR-Cas system and restriction modifications help protect bacteria from external DNA by lysing the specific DNA sequences. The detailed information in this article can give us a better understanding of how CRISPR-Cas system affects the evolution of bacteria, how the components of CRISPR-Cas affect plasmid replication, and how horizontal gene transfer is affected by it. Mechanisms of CRISPR-Cas is adaptation, maturation, and biogenesis, and interference. The process of adaptation occurs involving the acquisition of spacers. The DNA of invading plasmid is acquired for the CRISPR locus spacers. The region of the 5' on the CRISPR locus is where the addition of spacers occurs, which is usually derived from the fragmented or defective molecules. For spacer interference to occur, a protospacer adjacent motif known as PAM is needed. When the CRISPR locus adds spacer, it involves case 1 and case 2 proteins. Meanwhile, there is a duplication to preserve the organization of the repeat spacer region. The maturation and biogenesis of CRISPR RNA progress by the encoding of crRNA done through the repeat spacer region. The transcription of CRISPR locus occurs as a long sequence of nearly all CRISPR RNA, which is also known as pre-CRRNA. The pre-RNA is produced into short sequences of crRNAs along with the repeat sequences. The completion of the maturation of crRNAs is done by trimming at the 3' or 5' end. Interference develops as a result of cleaving the invading nucleic acid. The CRISPR-Cas ribonucleic protein complex form the core made up of the case proteins and crRNAs. An important factor of these complexes is to control the system that aids in immunity against the foreign invasion of DNA or RNA nucleic acid molecules that are complementary to every specific crRNA. This interference step is done by the recognition of a corresponding target sequence of either a viral DNA or a plasmid cleaved in a specific sequence. The recognition of PAM and these base pairing of about 8 nucleotides identical to the plasmid or viral targeted protospacers done by the crRNA spacer regions are an effective combination to activate nucleic acid cleavage. To avoid immunity, crRNA case complex are paired with nucleotides outside the PAM. This is how CRISPR-Cas systems target and recognize invading nucleic acid molecules, which help avoid the decimation of its own bacterial genomes. The horizontal gene transfer mechanism is prominent for bacterial evolution. The result of horizontal gene transfer relies on the nature of DNA being transferred as well as the host genetic background. The DNA that can be favorable under certain environments cannot provide a selective advantage to some bacteria that are usually grown under conditions that were different, which may decrease the fitness of the host. Bacteria have acquired a system to control the new genetic material from entering by blocking the entry of foreign DNA or by discarding mobile genetic elements from the bacteria cell. Bacteria are protected from foreign DNA by CRISPR-Cas and restriction modification systems that cleave specific DNA sequences. These two systems are cooperative in the bacterial cell and truly protect the bacterial genome from plasmids and phages. Through the mechanisms of natural transformation, transduction, and conjugation, the plasmids enter the bacterial cell, and once they enter the bacterial cell, single-stranded DNA is normally converted into double-stranded DNA molecules, which are then integrated into the chromosome or sustained as plasmid. If a protospacer crRNA sequence complements a new double-stranded DNA molecule, the CRISPR-Cas machinery will cleave it in a specific sequence. When an article transformation occurs through electroporation or heat treatments, double-stranded DNA can directly enter the cell. The CRISPR-Cas ribonucleic complexes can precisely target the double-stranded DNA molecules to remove them. There are few CRISPR-Cas systems that cleave RNA molecules. Meanwhile, the transcription of plasmid genes is silenced due to the CRISPR-Cas system, and subsequently, replication occurs for a few rounds and those plasmids can be lost. So this wraps up our presentation. Here is the list of our outside sources. And thank you everyone for listening.